So here's problem number one. It says below is a pedigree of a rare human skin disease. Yeah, this whole thing. And so basically the circle means female, the square means the male, and then if it's shaded in, it means that they're affected with the disease.
Okay.
And then, so inheritance of the disease by the 113 male right here from his father rules out what kind of inheritance. So there's 4 types. I'm just going to write them here. Autosomal, dominant.
And what's autosomal refer to?
Autosomal refers to basically any gene that's not sex-linked, that's not linked to the other. Autosomal.
And what is a sex-linked gene?
Recessive. A sex-linked gene is a gene that's carried on the X chromosome of one of the parents.
Okay. Only the X?
Yes. And then X-linked. Dominant. X-linked. Recessive. Okay, so an autosomal dominant would look like this. Say it's heterozygous.
Okay.
And so if the father was this, and the offspring, the male, could inherit any combination of genes from both of the parents. So it's possible that the offspring could inherit the dominant gene from the father, and then the recessive gene from the mother, and it'd still be affected.
Okay.
So we can't completely rule that out. And then autosomal recessive. Still, if the mother is recessive, she still wouldn't be a carrier of the disease if the disease is autosomal recessive, and the child could still inherit both recessive genes, and he would be affected too. For X-linked dominant, it'd be carried on the X chromosome. And the mother would not be able to have either of her X-linked genes be dominant, because otherwise she'd be affected. And the son would have to inherit the Y chromosome from the father, because all males have Y chromosomes. So he would have to have the recessive chromosome, and therefore would not be able to be affected. So it rules out this one.
Okay.
Next is X-linked recessive. So if we assume the mother's heterozygous again, which is like a worst case scenario, then
What's another option?
The other option is that the mother would be homozygous dominant.
Okay.
And so if we assume that the mother's is heterozygous, then obviously again the male would have to inherit the Y chromosome from his father, and then he could still inherit this one from his mother. It's still possible, so he could still be affected in this case. So between all these, the only one it rules out is X-linked dominant.
Okay, great.
Okay. And then who in the pedigree has the same 4 chromosomes, has the same Y chromosome as the 113 male, which is right here.
All right.
And so in this case, all of the males that are related to it will have the same chromosome, directly related. So this one, its father will pass it down. So that's 1, 2. Then as we go down, he's going to pass it down to 3, 2, because it has that as an offspring.
Okay.
And then these are females, so it won't pass down the Y chromosome. This one's unrelated. The next one is 3, 6, another male offspring. And then that's it from this row. Then
What does a row represent?
A row represents a generation. Okay. Yeah. So then 3, 2, this male will again reproduce, which is the only one that reproduces, which has the Y chromosome passed down. And then it goes to 4, 2 because these are all its other offspring are female, and then again it's passed on to another male. So those are the ones that it's related to.
Great.
Okay.
Yeah, that's good.
All right. So this is the second problem. The DNA content of an individual cell undergoing meiosis can be measured by applying a DNA-specific dye to the cell and then passing it through an instrument that measures the staining intensity. The more intense the sample is, the more DNA it contains. A new cancer drug was tested on a population of rapidly dividing tumor cells, and their DNA contents were analyzed and compared with those of untreated cells. Here's the DNA intensities, and this is the percentage of cells in each corresponding to each DNA intensity by untreated and treated.
Okay.
And so the first thing I did was use the sentence that I underlined: the more intense the sample is, the more DNA it contains. And actually, let's just start with the first question. So,
sure.
Plot percentage of cells versus dye intensity for the treated and untreated cells. Label the axes and indicate treated and untreated samples on the graph. So it's just a graph.
Okay.
And then the percentage of cells would be the y-axis. And then the x-axis will be the dye intensities. And then I'll differentiate between untreated and treated by the color.
Okay.
So dye intensities are 15, 30, 45, 60, and 75. And then the percentage of cells is going to go from 0 to 100 by tens.
Okay.
Yeah. So for treated cells, there's corresponding with dye intensity 50, there's 5% of cells in this. So, then corresponding with dye intensity 30, there's 60%. Sorry, there's 2%. Dye intensity 45, there's 0. Dye intensity 60, there's 93%. And dye intensity 75, there's 0%.
Okay.
And yeah, so basically I just did a bar graph because it would be all weird if there was a line, because there's pretty much 0 here, and then it shows that there's 93% in that area.
All right.
So for untreated, again we're going to start off with 5% and 15. 30 has 60%. 45 is going to have nothing again. 60 is going to have 35%. 75 is going to have 0. So, yeah, basically that's the graph showing dye intensities with percentage of the cells that are in those stages. Yeah.
Okay.
And then the second question is, at what stage of the cell cycle does the drug act on tumor cells? Let's see if I have this. Change this camera angle. There you go. At what stage of the cell cycle does the drug act on tumor cells? So the first thing that I did when I looked at this was just determined the DNA content per stage.
Okay.
So D1, it'll just be like, it'll just be like 1 times what it normally is. So that's like sort of the baseline. Then S, the DNA duplicates, so it'll be twice as many as in G1. G2, there's still going to be twice as many. Prophase 1, metaphase 1, anaphase 1, 2. And then after telophase 1, it's going to split up. and it's going to go back to 1 times as many because the cells have divided and essentially have their DNA content
Okay.
as before. So it's still 1. And then in the second stage of meiosis, they're going to divide again into 4 daughter cells. So it's going to be half as much DNA content as it started with, and a quarter as much as there is after it replicates, after S phase. So at what stage of the cell cycle does it act? So the first thing that I looked at, since the treated cells are the ones that are acting, I corresponded the treated cells here, since 93% of them are corresponding to dye intensity 60.
Okay.
I figured that they would be acting in dye intensity 60 because that's when they'd be used and they'd stay in that phase.
And what do you mean by that?
So they would be acting in this area when the DNA is proportional to that number, to 60.
Okay.
So then, since it says the more intense the sample is, the more DNA contains, this is the highest amount of intensity, and therefore I figured it'd be the highest, it corresponds to the highest amount of DNA content in the phases
Okay.
that I have right here. So,
I gotcha.
And the reason why 45 and 75 is 0 is because I figured that this would be the base amount, which would be in G1, which would be 1 times. You never get 1.5 times that in any stage of the cell cycle, and then you never get, 2.5 to 2.25 times that either. So all you're going to get is 1 times, 2 times, and 0.5 times. 0.5 is during cytokinesis, after cytokinesis too. And so when it says at which stage of the cell cycle it does it act, since 93% are treated acting in 60, and 60 is the highest DNA content when it's 2. I thought it would be acting starting after S phase ends, so end of S, because that's when there's 2 times the amount of DNA concentration.
Okay.
And then it would go from there to telophase 1, which is when right before it splits again.
Okay.
Does that make sense?
Does to me. And then why then do you have a couple here?
We have a couple here because they're still in the cell cycle. I don't know, they're just going until they're going to reach this phase and then they're going to act here.
And then with that makes sense. So yeah, no, I think you explained it well. Actually.
Okay. All right.
Graph looks good.
Yeah. Okay. Thank you.